So it's a very promising venue of progress. Given that we are using Brentuximab, which is the standard therapy that targets CD30. CD30 is universally expressed on the Hodgkin lymphoma cells. Since we're moving Brentuximab up in treatment, and a lot of patients are getting now in first-line therapy, we are facing, of course, the problem of how to target CD30 in patients who've already failed Brentuximab Vedotin. When these patients also fail checkpoint inhibitors, and most of them will eventually fail both, this is a population with a large unmet need. So NK cells are very promising cellular therapy because these cells can distinguish malignant cells from normal cells. So they can be used, you can infuse allogeneic NK cells to patients with, in theory, not encountering any graft-versus-host disease. And that is a big difference from allogeneic T cells. And in fact, we didn't see any GVHD in our study. My colleague, Katie Rezvani, has first published the trial of CD19 CAR NK cells against B lymphomas. And the her results in that population is very high activity and very safe as well. Now, with the off-the-shelf molecule AFN13, we can create a CAR-like phenotype in the NK cells without the burden and the cost of creating a CAR product, which requires gene transduction and layers of complexity in the lab. So I think this study will break ground for future trials where using different molecules, different innate cell engagers, targeting each one of them their specific tumor target, we can engage NK cells in fighting those tumors. In this case, AFN13 targets CD30, which is present on, obviously, CD30 lymphomas, but there are other antibody constructs binding CD16A NK cells and other tumor targets, such as BCMA, which is present on myeloma, or EGFR, which is present in myeloid tumors. And this constitutes, I think, a very promising venue, not only for lymphomas, but for many other types of tumor.